A single typographical error in a person's DNA can systematically destroy their vision. A standard eye chart, however, often fails to detect the collapse until the damage is advanced. Standard visual acuity tests focus on the macula, the center of the retina used for reading and sharp focus. Because many genetic conditions spare this area while the periphery silently collapses, these tests are structurally inadequate for early monitoring. This diagnostic blind spot means patients often endure years of misdiagnoses. By the time the condition is identified, years of psychological adaptation and critical windows for emerging treatments have already been lost. Detecting the threat early requires looking past the letter charts and examining the hidden electrical architecture of the cellular retina itself. Modern diagnostics bypass subjective vision tests to map the exact topography of retinal damage at a microscopic level. One of the primary tools is fundus autofluorescence imaging, which uses light to identify hidden decay by highlighting areas emitting an abnormally bright, hyperfluorescent glow. These bright clusters represent cells crying for help. These markers highlight where photoreceptors are actively degenerating due to a toxic accumulation of a pigment called lipofuscin. Using optical coherence tomography, clinicians inspect the ellipsoid zone, the second hyperreflective band embedded deep in the tissue. Its thickness corresponds to mitochondria health. Measuring this width provides a definitive biomarker for tracking disease progression. This line graph illustrates electroretinography, which measures the electrical activity firing across the neurosensory retina. When 30 to 40% of the retina is damaged, the electrical response drops toward zero, indicating a failure of full field function. Combining these three data points creates a precise coordinate map of cellular death. Establishing this baseline is the mandatory first step before attempting to intervene. In Leber congenital amaurosis, this failure originates from a biallelic mutation, an error inherited from both parents, in the RPE65 gene. This specific mutation halts the production of an essential isomerase, effectively starving the photoreceptors of 11-cis-retinol, the chemical fuel required to process light. Luxturna was designed to address this deficiency. It is a highly targeted genetic therapy with a current cost of $850,000 per eye. Delivery requires extreme physical precision. A surgeon uses a microsyringe to inject a targeted viral vector into the subretinal space, directly beneath the dying cells. Once the modified virus enters the cells, it unloads a wild-type gene sequence. The cell adopts this new code and resumes the normal production of isomerase. By restoring the internal production of 11-cis-retinol, Luxturna stops the cellular decay and recovers visual function that was previously lost. While the RPE65 mutation accounts for less than 2% of retinitis pigmentosa cases, its successful treatment demonstrates that human gene therapy in the retina is viable. This validation has accelerated phase 1 through 3 clinical trials for a range of other genetic targets. Research is currently underway to apply similar viral vector delivery systems to conditions like choroideremia and achromatopsia. Parallel research into direct DNA editing is also advancing. These therapies require deployment in the early stages of disease before the permanent loss of the peripheral rods and cones occurs. This comparison highlights an end-stage retina. At this point, the outer nuclear layer is devoid of cells. When light perception is completely lost, clinicians pivot toward hardware solutions like retinal prostheses or optogenetics to artificially bridge the gap. Advanced diagnostics and targeted viral vectors have provided a direct path to intervention for inherited blindness. By identifying and correcting specific biological errors, medical science is now preserving sight for patients who once had no viable options.